The heart is comprised of two main tissue types, cardiac muscle and the connective tissues interspersed within that cardiac muscle. The cardiac muscle is actually further subdivided into two main types of cardiac muscle cells. The nodal cells, which comprise the conducting system, shown in yellow here, and the cardiomyocytes, which are the contractile cells of the heart. Now note, the cardiomyocytes far outnumber the nodal cells. The nodal cells are non-contractile, and their function is largely to generate the stimulus for the cardiomyocytes to contract. They both generate that stimulus and then direct it along certain pathways to initiate contractions in a particular sequence. For instance, we would like the atria to contract and help fill the ventricles before they contract. So they have to contract first, then the ventricles have to be stimulated and contract. This is accomplished by the sequential traveling of this depolarizing signal amongst these conducting system cells, many of which we'll talk about a little bit later, like the SA node or the AV node. Now, it's important to note there is an electrical separator between the atria and the ventricles. It's called the fibrous skeleton. It does help stabilize the heart valves. You can see all the heart valves lined up along this pathway. But this fibrous skeleton is also electrically separating the atria from the ventricles, generating two functional syncytia. Now, a functional syncytium is a group of cells that are connected in such a way that they act as a functional unit. Now, cardiomyocytes are connected to one another, including the nodal cells, in fact, are connected to one another via gap junctions and desmosomes. This happens at the ends of the cells in a region called the intercalated disc. Now, the desmosomes are a mechanical connection. This is what allows the cardiomyocytes to pull against one another when they contract and not come apart, generating forces that will transmit through the walls of those heart chambers. The gap junctions are adjacent pores in two connected cells, and those pores dock such that the cytoplasm of adjacent cells is connected. What this does is it creates an electrical syncytium where depolarization can travel from cell to cell. In other words, current can travel from one cell to the next, initiating an action potential. That cell will then depolarize further and have current flow to another adjacent cell, depolarizing it. And so we get these domino effect of depolarization traveling through these functional syncytia. So the atria are in a functional syncytia, the ventricles are in a functional syncytia. You'll note our SA node is the starting point for the stimulatory depolarization that's going to generate a heartbeat. It's going to travel through those conducting system cells, those nodal cells throughout the atria, generate their contraction, which help to fill the ventricles. Then our fibrous skeleton, which has no gap junctions, right? It's limiting we can't just passively have our action potentials travel into the ventricles. And so it can only pass through at one spot, right? And it's right here. This AV bundle is a series of connected cells of the conducting system that pass through the fibrous skeleton. This allows us to have control of that stimulus arriving from the atria, generated in the atria, into the ventricles. And so that actually allows the atria to contract first. Then the ventricles are stimulated to contract. They generate their pressure and eject blood off into the systemic and pulmonary circuits. So this conducting system is absolutely essential to generating the intrinsic spontaneous heartbeat. In addition, the fibrous skeleton is absolutely essential to that because we need this separation of atrial from ventricular contractions in time, right? The atria have to help fill the ventricles before the ventricles do contract.